In the practical course of MB 3507, practical course 1, that is applied microbiology 1 for first semester, we'll be discussing about the different types of tests which are used for testing the quality of milk and milk product. We all know that milk is considered as complete food due to the reason of being nutritious because it contains carbohydrates, fats, proteins, minerals, vitamins, etc. etc. So almost all the essential components are present in milk, and therefore it is nutritionally rich that can support the growth of any type of cell. And therefore, due to its nutritious nature, milk also supports the growth of microorganisms. And if microorganisms are growing in milk, then it brings undesirable changes, and that leads to the spoilage of milk. Therefore, the purpose of all these tests is to determine the microbiological quality of milk, and second, to determine the compositional quality of milk. Now, there are several reasons for testing of milk. Let me explain you this very briefly, that periodic milk testings are required, milk analysis are required for basically microbiological analysis and compositional analysis. So these tests are to be carried out on raw milk, that is directly collected from the animal, and processed milk also, to check whether it contains any spoilage causing microorganisms as well as whether it contains any disease causing microorganism that may be responsible for zoonotic infection. That means sometimes the pathogens may get transferred from animals, infected animals, to humans through this consumption of milk. So that can be also tested, and that is one of the reasons for testing of milk. Then analysis of milk, it is carried out using standard techniques that provides the information in relation to the compositional quality of milk. Now all these milk testings are done to know the following things: whether the sample has been willfully adulterated or not. So adulteration of milk, it can be tested. The sanitary conditions of sanitary and hygienic conditions of prevailing during production and handling. So the extent of contamination, it has to be checked. Defects in product due to microbial growth or microorganisms responsible for it. Then presence of specific pathogens or toxic agents. Effectiveness of the methods of processing and preservation. Like we can check whether one of the best methods for milk preservation is pasteurization. So we can check whether the milk is properly pasteurized or not. So that is the test for processing and preservation. So in all, these milk testings will be divided into two categories: tests to determine microbiological quality of milk and tests to determine compositional quality of milk. Compositional means the major components present in milk are carbohydrates, fat, and protein. So these tests are based on measurement of carbohydrates, measurement of the content of fat in percentage, measurement of content of protein. So on the basis of that, the latter one, that is compositional and microbiological quality of milk. So we'll be discussing about both these aspects. So first in that, we'll be discussing about phosphatase test. So this test is responsible for checking the efficiency of pasteurization process, means whether milk is properly pasteurized or not. So this check is done. We can categorize this under microbiological quality of milk, because this is something which is related to microbial contamination and its destruction by pasteurization technique, that is uh, exposure of milk to high temperature for definite period of time. So the aim of this practical is to check efficiency of pasteurization of milk by phosphatase test. Phosphatase, what is it? It is an enzyme, and this enzyme is naturally present in living cells. That means it is naturally present in raw milk. That is the secretion of udder cells of milking animals. So it is naturally present in raw milk. Now, whenever pasteurization is done, the milk is exposed to high temperature, that is 63.6 degrees Celsius for 30 minutes, and at this temperature this enzyme is destroyed. Now, if milk is properly exposed to this temperature, phosphatase enzyme will be destroyed. So destruction of this enzyme, it indicates that the milk is properly pasteurized. So the presence of this enzyme indicates either faulty pasteurization or subsequent addition of raw milk. So it also indicates adulteration of pasteurized milk. So these two things are there. This test is performed to determine faulty pasteurization or adulteration of pasteurized milk with raw milk. So the amount of phosphatase present may be easily and quickly measured by colorimetric estimation. So this is what we are going to determine. This is the uh, basic introduction and the purpose of this phosphatase test. So here the phosphatase enzyme, it is used as an indicator. So if it is destroyed, pasteurization is good. If it is not, then it indicates faulty pasteurization. Now let us see the principle of this test. So it is based on the change of color. Now this test, it is based on the property of the heat labile enzyme, that is phosphatase enzyme, which is present in raw milk. And this enzyme, it liberates phenol from disodium phenyl phosphate. Now this disodium phenyl phosphate, so as it is an enzyme, we all know about enzyme catalyzed reactions. So whenever there is an enzyme, there is a substrate. The enzyme, it combines with substrate. There is formation of enzyme substrate complex. Then there is formation of enzyme product complex. Enzyme separates from product. So here the substrate for phosphatase enzyme used in this test is disodium phenyl phosphate. So this enzyme, it catalyzes the conversion of disodium phenyl phosphate to phenol. This phenol is a yellow colored compound. Now the color of milk is white. Phosphatase enzyme, when it acts on disodium phenyl phosphate, it leads to formation of yellow colored phenol. Now presence of yellow color in raw milk or the white colored milk, it is very difficult to detect, because white color with yellow color, the contrast is not good. In order to detect the change in color, we need a dark color. That means there has to be a contrast, increased contrast. So due to this, the next reagent which is added to increase the contrast is addition of this reagent which is written at the bottom, that is CQC. CQC stands for, it is a chemical 2,6-dichloroquinone chloramide. So this CQC, it reacts with this phenol which is liberated from this substrate, and it gets converted into a blue color compound. That blue color compound is called as endophenol blue. Now this blue color is clearly visible, so the contrast is very clear. We can easily detect the formation of blue color in white color milk compound. So this is the principle. Phosphatase enzyme, it converts this disodium phenyl phosphate which we add to the test tube. It gets converted to phenol. Then phenol gets converted to endophenol blue due to the addition of this CQC reagent. So the requirements for this practical are pasteurized milk. Then instead of pasteurized milk, we can make use of boiled milk. Now why pasteurized milk and boiled milk? Because these two are going to indicate the negative control. That means this is pasteurized, properly pasteurized. That means phosphatase enzyme is destroyed. This is boiled. Obviously, phosphatase enzyme is destroyed. So both are going to use to prepare a control. So whenever we perform a test, we need to have a positive control and a negative control. So if the results are positive, how it should look like? If the results are negative, how it should look like? So in order to compare your test with controls, you need to have a positive control and negative control. So this pasteurized milk or raw milk, it is useful for the preparation of a control. Then we need to have another control, and that control is, now here in both these milk samples there will be absence of phosphatase. Now we want one control in which phosphatase enzyme is present. So what is that? It is raw milk. So raw milk will be useful for the preparation of positive control in which phosphatase enzyme is intact. So this will be useful for preparation of negative control. This is for positive control. Then you will have a test milk sample. So in all, how many milk samples you have? Pasteurized milk or boiled milk for the preparation of negative control, raw milk for the preparation of positive control, and test milk sample. Then next is the substrate for phosphatase, that is disodium phenyl phosphate. It can be called as phenyl phosphate disodium salt. So this chemical is useful for the preparation of a solution called as buffered substrate. As it is related to enzyme, therefore this chemical is prepared in buffer, and therefore it is called as buffered substrate. Then 2,6-dichloroquinone 4 chloramide. This is CQC reagent. It is prepared in ethanol. So it is prepared as 1% solution. This is the reagent. So this is substrate, this is reagent. and of course 3 clean test tubes. So let me repeat the requirements: pasteurized milk sample or boiled milk sample to prepare negative control, raw milk sample to prepare positive control, test milk sample, test tubes, buffered solution of disodium phenyl phosphate which acts as a substrate, and CQC reagent. Now the procedure. So you see these are the 3 test tubes. So in these 3 test tubes, we have to take 4.5 ml of buffered substrate. Okay. So these 3 tubes are labeled like this: negative control, positive control, and test. So accordingly we need to make additions. So initially there is addition of 4.5 ml of buffered substrate in all these tubes. Now then add 1 ml of pasteurized or boiled milk sample and raw milk sample to test tubes. Negative. Now we have to prepare negative control and positive control. So this is negative control, what I told. In negative control we have to add a milk sample in which phosphatase is already destroyed. So it is pasteurized milk or boiled milk. 1 ml of pasteurized milk or boiled milk is added here. So that will form a negative control. This is positive control. This means that we have to add 1 ml of raw milk in which phosphatase enzyme is present, and in this test tube we have to add 1 ml of test milk sample, means the milk sample which is under test. So we have negative control, positive control, and test. So after addition, yes, pasteurized milk or boiled milk in negative control, then raw milk in positive. Then the contents are mixed, and then these tubes are kept in a water bath which is adjusted at 37 degrees Celsius, and it is incubated for 10 minutes for the reactions to happen. After incubation, the next step is addition of 2-3 drops of CQC reagent. So 2-3 drops of CQC reagent are added to all these 3 tubes. And then again the contents are mixed. After this, let us see how to read the results. Now these are your 3 tubes. Now if the results are like this, now you see negative control, it is boiled milk in which phosphatase enzyme is already destroyed. So obviously no reactions are going to happen, and this milk sample, it remains unchanged. The color remains white. So that is your negative control. Now here we have added raw milk sample in which phosphatase enzyme is active. So phosphatase enzyme, it will convert disodium phenyl phosphate to phenol, and when we add CQC reagent, CQC reagent will react with phenol to form to form indophenol blue. Now this is the color of the solution. It looks like this, and this is your test milk sample. So you have to compare the color of this test milk sample with these two. If the color is blue, it indicates that phosphatase enzyme is present. If the color remains unchanged, it indicates that the phosphatase enzyme is absent, and this is related to efficiency of pasteurization. So no change in color, it indicates phosphatase enzyme is absent. Presence of blue color, it indicates that phosphatase enzyme is absent, and here presence of blue color, it indicates that phosphatase enzyme is present. So present and absent. So see, these are the two conditions. Phosphatase is absent, means here. So if the results are like this, then you can conclude that the milk under test is pasteurized properly, or it is not adulterated by raw milk. You can write like this. You can add that a line regarding raw milk adulteration. Presence of blue color indicates the reaction enzyme catalyzed reactions have happened. So it indicates that phosphatase enzyme is present, and therefore the conclusion is milk under test is not properly pasteurized, or it is adulterated. It may be adulterated with raw milk sample.